Thank you for the invitation. Very much, because this is only the second time something like this. I hope I will not disappoint you. I select the topic that might be interesting for you. Hopefully. Let's see. Are you familiar with the concept of ecological stoichiometry? I know Cora is, but someone else, can you raise a hand who is familiar? So I assume that no one of you is, so I will introduce very briefly some basic concepts to set the background for the future theory that we developed so far. So ecological stoichiometry is something like chemical stoichiometry. The only difference is that we apply it to organisms, not chemical compounds. And I explain this by, chemical equation, which is very commonly known, I guess. At least most of you probably know it. Calcium oxide, water creates calcium hydroxide. Okay. And let's consider these as reactants, which are in the environment. They somehow interact together to form an organism. Okay. And now, when the composition of the organism is invariant, that means that it's always calcium hydroxide, it's not something else, then simple mass balance equation holds. And that means that, simply when you have some initial amount of reactants, they will disappear at some rate, depending on their ratio. Here you see that it's 1:1, and at the same rate, the calcium hydroxide will be formed. When you have no water, less reactants, you cannot expect that any product will be produced. And if you have excess of the water, you may notice that some of the water remains in the environment, in the solution, unreacted, because there is no contact. So you may notice from these graphs that what the ecological stoichiometry is good for, in most of the cases, is to guess new equilibria that will be established after some time when you know the initial conditions. You may also guess something about the reaction rates, not absolute, only relative, when you compare one to another. If you need to know something absolute, then you need to know the kinetic constant, which is something more difficult to get. And Yusuf Omid is the founder actually of the ecological stoichiometry, because he was the first one who applies these known principles from the chemistry to biological system. Not surprisingly, he was a chemist, so that was a natural thing to do. And he defined the Liebig's law of minimum. You already probably know it. And it says that the yield of production of plants is determined by the nutrient which is the most used. And how we work with this, simply you can make stoichiometric ratios or ratios of any two, even more elements, and compare it to each other, between environment and organism. And so, for example, that the organism here has a ratio 1 to 2, 0.5. And that in the environment, you have this ratio 1. And when you compare these two, you immediately can say that the hydrogen will limit the yield of the reaction. Vice versa, if you have something different, lower than this ratio, that calcium will limit the yield. This is a little bit tricky because, I noticed that thinking in these ratios might be tricky because you have to realize which is the numerator and denominator, and sometimes it can create problems. Another thing is that you have to look at the nutrients that are important. And in terrestrial ecosystems, so far, we believe that especially nitrogen and phosphorus are the two that are the most important. And people around the world do fertilization experiments to find out which one of those is limiting the yield, okay? And they express their results typically in something that is called log response ratio or a response ratio. And this is calculated by this simple equation, plant growth following either nitrogen or phosphorus addition relative to control. It means that when you have the log response ratio on the y-axis, okay, it's a log response ratio. So if the ratio here is 1, logarithm is 0. So when the log response ratio is higher than 0, that means positive, or and vice versa, negative, when the plant grows less. And in most of the cases, or for a very long time, it was believed that most or almost all terrestrial ecosystems are limited by the nitrogen. If this is the truth, we should see something like this. We are adding nitrogen, the plant growth increases. We are in phosphorus, nothing happens. You know, plants are not limited by the phosphorus. If we add both, then the increase need to be positive, and it must be higher than the addition of only nitrogen. This applies from the mass balance equations that I just showed you before. This is very simple. And now, just before I go ahead, I just have to make a note, because this is something people make mistakes in, and it's almost everywhere. And if you read papers about ecological stoichiometry, you have to be aware that there are two different ratios that differ to each other quite significantly: mass ratios and molar ratios. Some scientists use mass ratios, some scientists use molar ratios. And that makes a difference, because when you make a ratio between nitrogen and phosphorus, with 10 micrograms and 1 microgram, for example, you will get the ratio 10. And on molar basis, you get the double, because the phosphorus is very heavy. Simple. And we typically use molar ratios, but other scientists use mass ratios, then they compare their ratios, and they are often in the disagreement which is only applied. Now, stoichiometry of nutrient mineralization in the soil. I'm a soil scientist. This is my background. And we apply similar stoichiometric relationships to what works, how soil works, or that is to say, how microbes works in the soil. Why we do it? Well, this is a table that shows you that across most of the ecosystems, doesn't matter which one it is, the new inputs of nutrients are relatively small. So more than half of all nutrient supply that plant take over the year gets from the mineralization of organic material that is in the soil. So this is significant. And now I will go very simple scheme to explain how it works and how the ecological stoichiometry applies here. So we have pools, organic compounds, and microorganisms per carbon basis. Microorganisms consume organic compounds, and they either respire or grow. Assuming we have heterotrophic microorganisms living at aerobic conditions. The microorganisms that are there sometimes eventually die and becomes organic compounds again. Then there is one very important parameter that is called carbon use efficiency, that defines how much of the carbon taken up by microbes is allocated to the growth and how much is allocated to the respiration. And now each of the pool is defined by its stoichiometry. So let's write stoichiometry to respect any nutrient. It could be nitrogen, phosphorus, potassium, calcium, whatever, and denote the nutrient as an E. So we have organic sources with C to E ratio and microbial biomass. And now uptake of the nutrient can be right as a function of the uptake of the carbon and the carbon to nutrient ratio of the source. Okay. Demand for the growth of these microorganisms for this nutrient or let's say any nutrient is then defined as a function of the uptake, carbon sufficiency that says what is the allocation to growth, and the carbon to nutrient ratio in the microbial biomass. If the uptake is higher than the demand, obviously there is an excess, and the excess is mineralized. Okay, so mineral forms of nutrients are released to the soil, ammonia or phosphates, and this is what plants uses to grow. So they taken up, growth, and this is the mineralization that creates this 50 to 60% of the entire supply of nutrients to plants, whatever ecosystem it is. But there could be another situation when the uptake, or let's say demand, exceeds the uptake. If that happens, microorganisms have a tendency to balance the difference by taking up mineral form of nutrients. So nutrient mineralization is not sure thing. It happens only at specific conditions. Sometimes microbes mineralize, sometimes they uptake mineral nutrients. If they take up nutrients, they are actually in competitive relationship to plants because this is a shared resource. And how much the nutrient will be either mineralized or taken up can be written down by this very simple equation which just compares technically demand and uptake. And this is called, let's say, well, the flux is called mineralization, but it can be either positive or negative. So mineralization is technically only if this is positive. If it's negative, then the nutrient is taken up. And you can write this equation into respect to any two atoms or elements. So for nitrogen, it would look like that. For phosphorus, it would look similar. And you can also compare the nitrogen and phosphorus together in any way. The equation is very versatile and general applicable. Okay. And the point is that when plants and microbes share a common resource, the microbes are better competitors because of their physiology, cell size, distribution in the soil. So if you will compare plants and microbes will always win the competition. This is how it works. And plants probably quickly realize it, so they form symbiotic relationships together, and they help each other in symbiotic relationship to all. How important these relationships are shown on this global map. Just compare these two scales. This is for the direct root uptake. I think in this case, it's nitrogen, and the mycorrhizal uptake. So you see that these scales are quite different, and it's estimated that like 13% of net primary production is allocated to mycorrhizal symbionts. Now, and we are getting almost close to the theory. And before I go into the detail, I have to mention just a few things. When I did my PhD, I worked in Arctic. And my duty was to study the phosphorus in the Arctic, the cycling in the soil, how it works, how it affects everything. And we had brilliant PI, and he's like big scientist, great mind. And right at the beginning, when I met him, he told me, "Man, forget about the phosphorus because everyone knows the nitrogen is emitting, everything in the tundra." which was quite disappointing because, you are a PhD student, someone that you admire tells you this is what was to do for next 4 years. So it's like, and at that time I believed him completely, but I was suspicious. You should be always suspicious, even if something could be obvious and it's articulated by very good scientists, because sometimes even very good scientists are not. So the logical nature question was, okay, so how do you know this? How can you say that tundra or any ecosystem or microorganisms in any ecosystem are limited by the nitrogen? You will say, okay, because of this, right? Because you know that there are a lot of fertilization experiments in the tundra, and when you add nitrogen, it always increases. But microorganisms are not plants. So are you sure that microorganisms actually behave the same way? So they react in the same magnitude, in the same direction? And then I did my own research, and I found out this meta-analysis of all fertilization experiments that shows something quite opposite, or something a little bit strange, because they don't show this kind of picture. This is the response ratio plotted for different ecosystems. You can see that something that you won't expect is that nitrogen fertilization increases plant growth, phosphorus fertilization increases in the same magnitude, in the same direction, and this is logical. We don't need to pay much attention to it, but how this can be, because you cannot really, or plant cannot really replace nitrogen and phosphorus and amino acids. That's just impossible. So how it physiological works? If you read this paper, they provide kind of vague explanation about it. Okay, plants somehow affect their environment, so they are always limited at the same time by nitrogen and phosphorus, and this is what you should expect from that relationship, which for me at least was kind of unbelievable. And I was asking the question, so is the leading law of minimum relevant in this situation? Because it doesn't seem to me it's not. It should behave like that. It does not. And then when you look closer to the data, you may find out that in many situations, many ecosystems, when nitrogen is added, it increases primary production, but when phosphorus is added, the primary production decreases. And again, when you look at the explanation, it's kind of vague, because they say, okay, if you add something, in very high amount, or, in a form that is probably not very suitable, it might get toxic and the plants will grow worse. Which is, again, how much is much, and what is the correct form. So the question is why the nutrient addition decreases. And that brings us already to our theory about the plant-microbe interaction framework that actually explains all of that by very simple principle that says terrestrial ecosystems are not only plants, it's some of the plants and microbes that might interact with each other in different ways, in stoichiometry-wide ratios, and all these aspects we observe can be simply explained by that. So, but first we have to answer our questions as we raised. So are plants and some microorganisms limited by the same nutrient? If we express this graphically, we expect that, in this plot, the organism's demand, if the organism is chemically invariant, you should observe this line, which should be same for the microbes and plants. And then you know that here the organism is phosphorus limited and here is temperature. So we did a meta-analysis across all ecosystems, and we saw this. So here, it's not phosphorus concentration, nitrogen concentration. I will plot this scheme again, but I will first show you this graph. On x-axis, we have nitrogen to phosphorus ratio in the soil. On y-axis, we have something that we call nitrogen to phosphorus critical ratio. So this is approximation of the demand. So in this case, we plot simply nitrogen to phosphorus ratio of biomass of soil microorganisms. And here for plants, we plot nitrogen to phosphorus ratio in plant leaves, because those are, the metabolic centers of the plant. And this is one-to-one line. If the critical ratio follows one-to-one line, then indeed, organisms is always limited by the nitrogen and phosphorus at the same time. However, this doesn't work. Plants, to some extent, copy the changes in the nitrogen to phosphorus ratio, but only to some extent. Microbes do not. But most importantly, these two lines are shifted against each other. And when you look on this line, you can actually find that here both are limited by the same nutrient, in this case ratio. Here, when we are below that line, both are limited by the phosphorus. But there is a certain range of NP ratios when plants are limited by the nitrogen, whereas microbes are limited by the phosphorus. And it's logical, plants are different organisms, so you cannot really expect that they will have the same demand. They need less phosphorus than microbes. So this figure does not apply, this figure does. And, based on this, you can answer these two questions. Does the Liebig slope minimum hold true? Let's say it does. And why then the nutrient addition decreases? And why addition of nitrogen and phosphorus increases the plant growth at the same time? So let's look on that closer. We have both nitrogen limited, nitrogen phosphorus limited, and in many cases, for some microorganisms, happens that they are carbon limited. So this is the fourth condition that we have to acknowledge here. Now, those are the color codes for this part of the graph, and we can derive predictions for each of the situation depending on what nutrient you add and what is the relationship of plants with microorganisms under that scenario. So let's look on that as a background. So here, in this half of the graph, we have a situation when plants are in the competitive relationship with microbes. Okay, you add nitrogen, plants are nitrogen limited, they will grow better. You add phosphorus, that means that microorganisms will grow better. You switch the limitation from the phosphorus to nitrogen limitation, and you cause microbes to be in competitive relationship with the plants. And because microbes are better competitors, they will win the competition and steal nitrogen of nitrogen limited plants, so the plants will grow less. If, on the other hand, plants are in cooperative relationship with microorganisms, nitrogen addition increase, plant growth, phosphorus addition will increase as well, because when your symbionts grow better, it will provide more nutrients. Similar situation applies to shared nitrogen limitation. Here, you see that the situation is same. This might be kind of unlogical or not obvious at right away. But what you do is actually that you increase the phosphorus concentration here. So you decrease the denominator, and the mineralization becomes more negative. Okay, that means that when you add a phosphorus, you worsen the nitrogen limitation and increases the competition for the nitrogen at nitrogen limited conditions. And again, the phosphorus addition will cause plants to grow worse. And if plants and microbes are in the cooperative relationship, nothing happens after phosphorus addition because symbionts obviously don't want to harm the plants. So there will be something we expect for only plants based on the Liebig's law of minimum, not accounting for the microbes. Then we have the phosphorus limited situation when you can see that the effects are just the opposite. And then we have carbon limitation, and then it doesn't matter what nutrient to add, it will always increase the plant growth because what you worsen here is the limitation for the carbon. And if microbes are limited for the carbon and the limitation increases, they will tend to allocate more resources to decompose the organic material, and when organic material is decomposed, everything else gets minimized.
Another question. Isn't like the third option that is unlimited, but also like carbon limited, so that like microbes don't like follow with like growth, but like plants skyrocket because they utilize the free nitrogen and phosphorus?
Can you repeat it? I'm not sure I fully.
So that like you have this market, and like microbes don't get the carbon, so plants can utilize all the free nitrogen and phosphorus that is given to the system. So that's why it's skyrocket, and it's like different from the.
Yes, but at that situation, you might expect that it depends on if the plant is nitrogen and phosphorus limited. And then you cannot see, the same response. So you have to expect here that this is the increased mineralization at any condition, because if, for example, plants will be nitrogen limited here, then of course the nitrogen addition will increase plant growth. But if you add the phosphorus, then you cannot expect this. So you have to explain it by the increased, mineralization of the nitrogen by application of the phosphorus that is caused by the worsening of carbon limitation of microbes. It always takes time to digest it a little bit because these ratios are a little bit tricky, and there is you really have to think what is in the numerator and denominator and how it affects each other. But once you digest it, it's quite simple. Yes. Yeah, this is actually how you can explain it for nitrogen, but you can draw this same equation again for the phosphorus. So this was my hypothesis, and I decided to test it. I didn't decide to test it directly, that I will do experiment, because I thought that it's possible to just do meta-analysis, because as far as I know, there are like thousands of fertilization experiments. And this was, unfortunately, the most disappointing work on this paper which I ever did, actually, because there is a lot of fertilization experiments, but most of the data cannot be used. So we defined several criteria that the study must comply with to be included in the meta-analysis. Fertilization experiment must be factorial. This is, I guess, obvious. Initial conditions must be well defined, because only if you know the initial conditions and you know the additions, then you can guess the new equilibrium. Without it, this is not possible. For plants, the information are always available. For soil and microbes, almost nothing. Which is actually quite strange, because if you know that, plants take the nutrients from the soil, and the mineralization is very important. You probably want to know what is in the soil after application of nitrogen and phosphorus, and for some reason people don't know. Then plant and soil microbial characteristics must be ideally measured over time, so we can make sure that the equilibrium which we guess was really reached. Sometimes that was not possible, so we didn't strictly apply this rule. Basic soil parameters must be known because we have to distinguish between the nitrogen limitation, phosphorus limitation, or carbon limitation. And this was again quite tricky, and informative units must be provided, which is also not very often the case because sometimes, well, very often actually it was surprising that scientists measure total nitrogen and then some available form of the phosphorus. And from these two, you cannot, make informative ratio because you compare pears and apples. And sometimes, scientists provide units in like micrograms per liter without, mentioning what the liter actually means. If it's the volume of the soil or if it's the volume of the extractant they use to extract the available forms. And, reading through all these thousands of papers was really hard and disappointing. Plant and soil community composition should be known. This is required for us to recognize if at that specific locality plants more likely cooperate or compete. So from these thousands of applications, we were able to select 51, which is a very low number, but unfortunately, we cannot do much about it. And now, actually, this is almost close of my presentation. Those are the theoretical predictions, and those are the results. So you see that in most of the cases, the theoretical predictions fit what is observed across all ecosystems. Once I get the question, okay, so those are only agricultural ecosystems with only one plant? No, it's all ecosystems from everywhere, and including agricultural ecosystems too, with very rich sometimes plant community. And if you test, for example, the effect of the ecosystem or mean annual temperature or precipitation on these log response ratios, they cannot explain much. Most of this variability can be explained by this theoretical framework, except of two cases. And here, as you can see, expect that in phosphorus limited conditions, adding nitrogen will either have no effect or decrease plant growth. The opposite actually happened, which was not very surprising after all, because there is one more important thing we didn't consider at the beginning, and that is that either plants and microbes to get nutrients from organic resources produce extracellular enzymes. And extracellular enzymes are, amino acids, proteins, and to build proteins you need a nitrogen. So when you add the nitrogen, you can trigger the extracellular enzyme production of phosphatases. There are meta-analysis on that, and it really works. So they get by additional nitrogen to the organic phosphorus forms. And that's it. Limitations. Every theory has limitations, so I will end my presentation here. The first limitation obviously is data quality, because the data quality is poor and it would be hard to do it better next time. It applies only to condition of nutrient limitation and specifically nitrogen and phosphorus, and we already know that not in every ecosystem only nitrogen and phosphorus have to be considered. In many ecosystems, calcium have to be considered or potassium. Parasitism is obviously ignored here. Sometimes symbiotic mycorrhiza can really behave as a parasite, not as a symbiote. And just one note, these ratios which we apply here usually define the global needs. So you saw that there is certain variability here. You should see gray points, small gray points. Can you see it? So those are the real data behind it. They are probably not very visible here, but it stems from here. Also in this condition, you can see the opposite results that cannot be explained at the moment by this theory, and maybe it's because of the data, maybe it's because something else, and maybe the ratios which we found did not apply because what we could really get from the papers was the total nitrogen and total phosphorus, and we would ideally need available forms of nitrogen and phosphorus, not the totals, because they are not correlated very well. And one thing I have to admit at the moment, this paper has several co-authors, and not all of them entirely trust this theory. So there is no place to improve it. Okay, thank you for your attention.
Thank you very much for showing that why our experiments do not work. Okay, and well,
Yeah, I have a question about how do you define whether in a given system the plants and the microbes are cooperating or competing?
So you either know the composition of microbial community, and you can calculate, for example, fungal to bacteria ratio, and you can know what proportion of the DNA extracted belongs to mycorrhizal species. If you don't know that, which is most of the cases, what we did is that we look at the composition of the plants, and there is a database of plants where it's specifically noted which one creates mycorrhizal relationship or symbiotic relationship with nitrogen-fixing bacteria and which are not. So if there were a lot of species with high abundance and high biomass that do not form these symbiotic relationship, we just consider that the plants and microbes are in the co-competitive relationship, which of course is not like super accurate.
But you didn't get different results in the places where the competitive and non-competitive and cooperative situation would give different results compared to when they give the same result, right?
Yes.
So in general, you managed to separate these groups reasonably well.
Well, when a reviewer asks, say yes, sure. More honest answer is I don't know. I think we try to be as accurate as possible, but you cannot really. And if you write authors of the paper, they usually do not respond.
so maybe I will ask a practical question. If I'm thinking about potential future fertilization experiment, calling to Pros and asking him how much of the nitrogen I should be pouring there is maybe not the best. So what should I measure in advance? So the nutrient content, how do I kind of proceed?
So ideal is to measure nutrient content, not the total amount, but some available forms. Usually we do the extraction with water. This is the most labile form of nitrogen and phosphorus. And ideally also to measure the nutrient content of microbial biomass and the composition of the microbial community.
Okay.
So this is the DNA extraction, sequencing, and something that we call fumigation extraction method. That is used to measure the content of carbon, nitrogen, and phosphorus in the entire community of microbes in the soil.
And after that, I need to know whether the plants on the plot which I will be managing are microbial or not, mycorrhizal or not.
Yes,
And after that, I can decide how much I will
Yes.
be fertilizing.
which I understand is demanding, but if you really want to get roughly correct answer, I would vote for do it.
Mhm.
But I understand this is time demanding and financially demanding. That's,
Yeah, but then you might be wasting several years of fertilization as we did, or maybe, because we don't know what happened there, and yeah, see no results because maybe you were doing something wrong.
well, and then for sure, it also depends on what form of fertilizer you apply, because sometimes the loss of the fertilizer can be significant, especially if you use ammonia, that is can be volatile quite quickly, or if you apply nitrate, it can be nitrified. You are losing significant amounts of the especially nitrogen fertilizer. And if in respect to phosphorus, if you have phosphorus poor soil, there will be high absorption of the phosphorus to clay minerals, and what you add is not available. That's why you probably need to know the available forms of phosphorus.
So the related question to that. Okay, so if we have some kind of step-by-step guide how to go through these steps, how the sequencing of bacteria will help us. We should arrive at kilograms of whatever form of chemical form of nitrogen and phosphorus we want to apply for some sensible experiment. So how this bacteria information will be used for that?
Well, that information can be only used to guess if there is a competitive or cooperative relationship, not for the amount of fertilizer that has to be applied. Then you have to follow only the available forms.
And this absorption of phosphorus and possible also leaking from the soil. I suppose there are some protocols if you want to analyze soil properties.
Yes.
To estimate it.
Yeah. So you can estimate the maximum capacity of phosphorus absorption that soil has, and again, it is kind of demanding, but it is manageable. And maybe also one important information. It often happens that when you apply a lot of nitrogen, plants will stop cooperate with their symbionts. It happens very often.
Yeah, that's I can imagine. One unrelated question about your meta-analysis. So most of these experiments were not well designed, or it was more like not the right kind of data were published, so that if you wrote to the authors, you might actually get what you need. I remember when we were writing a review on host specialization of herbivores, we did some analysis, and we pretty much had to write to almost every single author to get the information we needed. So
yes, the problem was in lack of the data that we need, especially the soil data and microbial data, and unresponsiveness of authors in this case. So I'm happy you had better experience. I did not. Some of authors wrote back, but they said, okay, we don't have data. So we also did a lot of gap filling, which we sometimes discuss in the detail, and they are also not super accurate, but at the end it made some picture. And in several cases, the data were obviously wrong. I had to say straightforward.
So who is doing this kind of research? Is it more soil biologists or more botanists who are somehow branching to soil?
Botanists.
Okay, because that looks like a law of upper trophic level. Like when you look at plant herbivore interactions, it's always zoologists who are doing that. Yeah, botanists will never think about what is eating their plant. So this is interesting that this is the same level on sorry. Yeah. I don't know about the predators, but maybe the same. But
Maybe because we are also torturing some plants by drought, how does the water availability can mess up these results? If there is some kind of simple answer.
So, yeah, so, well, the water availability in this study is assumed to be unlimited. If you limit the plant by the water, there is no diffusion of nutrients, and then you cannot see any effect of nitrogen phosphorus fertilization, because no matter what you add, it cannot really diffuse toward the because this is a diffusion limitation. There are actually models from that. This can be modeled.
One more question. We do also some experiment where we want to, kill, some organisms in soil. So we do fungicides with very shaky results, but that's another story. On the plants, we do insecticides. Are there any experiments which will actually try to kill the soil bacteria and see what will happen?
Some experiments do it, not field experiments, as far as I know. They are already quite old, from 97. 2 experiments that planted plants into pots with autoclaved soil, with all microbes killed, and not autoclaved soil, which was actually part of this meta-analysis as well. And in that case, it shows quite significant effect, because once you forget about microbes, then plants can behave exactly as the Liebig's proposed.
But doesn't the autoclaving change the composition of the soil?
It does, yes. It releases carbon, it releases nitrogen, it releases phosphorus, and authors of that study somehow acknowledge that, because they measured the available forms, and at that time they could define the relative increase above what was lost from the microbial biomass, because like in relative terms, microbial biomass, if you release all the nutrients from the biomass, it may be equivalent or even higher than all the available forms today.